screen now, which is a Zoom screen. And I thought I'd start tonight by going over some of the photographs that have been coming in. And guys, I really do appreciate them coming in because it gives us a chance to talk about the things that you're seeing and deal with those bits. So let's see if I can deal with that by. Okay, so can everyone see the image of the fungi here? Someone give me a thumbs up. Yep, okay, great. So Michele sent this one in. It's at the base of the tree. One of the things that we notice straight away is that there's live tissue. So we've got the bark being shed and there's live tissue in behind. So without the sporocarp being open more, so sporocarp, the part that bears the spores, that being open more, I'm guessing on this Angophora that is Ginopolis. And there's probably a little bit of dead tissue there. That's what that looks like based on that image. This doesn't look like Ginopolis. This looks like it smells like curry. So this is looks like a curry punk, which is a brown rot often associated with fire. Was this out when you're out doing surveying, Michele, or?
Yes.
Michele is frozen. Everyone's frozen. Oh no.
I can hear you.
Okay, good. So just Michele's frozen. That's fine. Vicky's just sitting there so still that I thought she was frozen as well. Okay. So this looks like the curry punk. It's a brown rot that's associated with fire damage as well. It's usually not a big problem for trees. The only brown rot failure that I've seen in the time that I've been working, one was on a uke and one was on a pinus, and the pinus was a pretty extreme situation. So brown rot's not generally going to be a big concern from our side. That's what that looks like as a thing. And I can't, Michele's dropped out, but the fact that he's out surveying, I know that he's out doing country areas at the moment, I suspect it's probably been an area where there has been at least a reasonable fire at some stage or other. So we'll find out from that one when he gets back on. So next image. Someone sent in these images here on the base of, looks like a poplar, and that was from David. David, are you there?
Yeah, hi, I'm here, yes.
Were they from a poplar?
That's correct, yeah, there's a row of poplars in near Mansfield, Victoria.
Oh, they're in a zip format. Okay, well, let's see if I can download those from a zip format. It may or may not let it work evenly and smoothly.
Yeah, so they noticed about 18 months ago that the trees were starting to decline, and they removed a few and yeah, asked for a bit of advice as to what might have been going on.
It was certainly, oops, that's not one. We've got a canker of some description, and a canker is just a nice technical name for an area of dead tissue, necrotic tissue, and it looks very much like that it might be something like a bacterial infection. We'll come in a little closer. And there are probably other reasons why the trees aren't going well, but it definitely looks like bacterial slime flux. Because there's food in the tree, the starch, some organisms are able to avail themselves of it. Occasionally bacteria do come in, they take the sapwood and they break that starch down. And when you take starch or sugar and you take and yeast fungi can do the same thing. You take the sugar and you start breaking it down, you produce ethanol and carbon dioxide. The carbon dioxide then forces fluids out. The end result of the process of fermentation is alcohol, and alcohol kills living things. That's why arborists drink, is to kill brain cells. Like, we need to do that. That would be a stupid thing, but it's certainly the joy of drinking is you get some in your blood system and some goes up to the brain, kills the brain cells, and gives you that momentary pleasure that you enjoy, for those of you who drink. That's what it looks like, bacterial slime flux. Generally, it's not a problem for the health of a tree. And, you know, when we talk about generalizations, we talk about, for example, an average being warm. Well, that could mean you've got 1 foot in boiling hot water and 1 foot in freezing cold water, and you're warm. No, it means that, you know, there is a distribution curve. So it could be a contributing factor, but normally not the cause of death. There are a few fungi that do cause bigger problems. Here, we've lost all our bark. So complete necrosis. And that doesn't seem consistent with what we're looking at earlier on. So I think they're two separate things going on at the same time.
Mark, if there are a number of things causing stress, then they kind of could be combining together. And
yeah, what we call a disease complex, yeah.
Yeah, one might not be the cause of the decline and death, but when you add them all together, they can, yeah?
Yeah, absolutely. And this has been discussed in great detail by people like Mannion, for example, with the disease decline, or the disease spiral. It's called all sorts of different things. It's a process of things getting, the tree being stressed or stress being induced by any one of a number of different things. And then the process not rectifying, you end up with something else taking advantage and something else taking advantage. And we're going to look at today at several fungi that fit into that category that generally don't do anything that's problematic until the tree is reasonably heavily stressed. So excellent. Thanks for those photographs. Let's go back to emails. Now, this is again from David, and this is a tree. Let me just blow these images up for you if I can. Maybe I can't. Yeah, I can. Good. Okay, so again, we've got this conch or this bracket fungus sitting on the side, and this one looks more like a whitey color than a browny color. The tree's had a number of failures, and I'm guessing by the looks of the size of it, is it a Pterocornis, David? Do you know? Sorry, Camargulensis?
Yeah, that's right. Yes.
Yeah, which shed branches occasionally in any case. So may or may not be related to the fungus that you're seeing, but that fungus looks like the white punk. It's a brown rot, and again, we'll go over that in a bit more detail later on this afternoon. Ladioporus portentosus, for those who want a nice Latin name. And it does occasionally lead to failures, particularly where it's located on just a limb, but on the stem, it doesn't often go that way. And there is, you know, when you're seeing a big lateral like this, there is a lot of tissue being laid down on the underside here, and that adds incredible amount of strength. So it's going to be depending on lots of things and how you'd respond to that. Where you could, you'd do some weight reduction, but you don't want to do too much because you don't want to take out too much canopy. The reason you don't want to take out too much canopy is we need canopy for food production, for growth, and the tree is best able to deal with these sort of problems by growing. So keep in mind when you want to do management that we want to try and reduce the amount of loss of photosynthetes whilst at the same time reducing load on those sort of things. So wonderful again. Thank you for sending that in. Now, last time around, I didn't get to address this one. And again, from David. David's got the message, send in images and we'll have a go at them and see how we go with getting them right or wrong. It's an abutus unido that has had a fairly big basal wound formed. David, what else can you tell us about the tree?
Excuse me. Well, it's in a fairly old garden over near Euroa in Victoria, and on one side there's, as you said, there's a big wound with quite a bit of decay in it, and the third photo there's a cavity. You can see sort of in the central trunk, which is up there. It's full of bees.
Okay,
So it's a
question:
how do hive in the tree?
Yeah, how do bees influence? Bees are really good housekeepers. So yes, what are bees like in trees? Bees are just another user of cavities. Most of the bees that we have, unfortunately, in cavities are European. It's not that I'm against European bees. I cop a bit of heat occasionally from some horticultural experts over my disrespect for them as a pollinators. There are lots of pollinators out there apart from bees. And I think sometimes we get caught in big, broad scale horticulture rather than thinking about some of the other pollinators. But they're great housekeepers. We don't have big problems with decay.
Mark, I sorry to interrupt. I haven't sent you the photos of it, but I did see some what I think are bumblebees pollinating flowers here the other day.
Yeah, and if you go down to Tasmania again, there somebody was actually caught a while ago breeding bumblebees to pollinate in Tasmania, and of course they're a prohibited plant creature, even though they're in Tasmania. You're not allowed to breed them up for that purpose. So, and of course we've got a great number of native bees that are pollinators. One of my favourites. One that nests in the ground, the teddy bear bee, and it's really quite a good pollinator. Many of our bees are. So they don't have to be colonal to be good at the job. But where they are, where we've got exotic bees, European bees, they tend to be good at looking after the cavities. And most cavities are desirable things. You know, we oftentimes think of cavities as negative, but trees don't often fail at the cavities, and when they do, they're in extreme winds. And it's amazing. A couple of the fatalities that I've looked at in the last decade have had trees with incredible cavities. So, for example, the tree that killed Bridget Wright, it had bees, it had a cavity, it had Phellinus. None of that failed. A branch failed. The Emu Plains fatality. The tree had Phellinus, the tree had cavities, and the only reason it failed is the tree beside failed during a microburst. So the cavities didn't fail. The Phellinus wasn't the cause of the failure. It was simply wind load on an adjacent tree and then the combined load being too much. So bees are generally not going to be a problem. The cypress and the cypress group. In this case, it's not a hedge, which is lovely to see. But we've got a tree that would be better suited to higher moisture levels. Now, who is this one from? Vicky. Vicky, this is one of yours. Two big problems that cause grief for us with cypress. One is cypress canker. That tends to be a problem associated with pruning and management. And the other one's cypress weevil. And cypress weevil is a native beetle, endemic to Australia. And every now and again, when we've got exotic plants and we expose them to our native insects, our native insects can cause damage to them they wouldn't be causing to our native trees. So Calitris, for example, will only ever be attacked when it's dying. But you'll see cypress weevil will come in and damage trees that are in medium health that are a cypress. And in the 1940s, there was a big population of Pinus halepensis in the Concord region, and indeed cypress weevil took those out. And it just so happened that the combination of the environment they were growing in and the numbers of them just became a problem. The weevil went through and wiped them out. Lastly, that's probably our bigger ones in terms of cypress. So the things that create issues with cypress, things like this driveway obviously is affecting some soil moisture ingress. And the leaves are waxy, which means the mulch underneath these things is almost always hydrophobic. There's an advantage to a hydrophobic mulch underneath a tree. It gets rid of competitors, but it also reduces water intake. And I've gone out and tested underneath a Cupressus at the University of Western Sydney after 100 mils of rain and still found no moisture had penetrated through the cypress. So that can be problematic.
Mark, if you wanted to check that it was the weevil attacking these trees, would you look for holes in the trunk?
Yeah, two things you look for. First of all is they chew the bark off, adults chew the bark off small little branchlets. So you'll see little dead tips, and they're a good giveaway that you've got the adults around. And then you'll see oozing from the bark. The tree will try and produce large amounts of resin. And if the tree was really healthy, it would flush out the young larvae, the young grub. But when the trees are stressed, they stop producing the defense system to the same extent. It's the same with eucalypts. It's why, for example, after heavy rain, you'll see lots of kino being produced by eucalypts. The kino is produced because all of a sudden the water's got the system going again, and we can spend water. Water is an expensive chemical to produce, to get, to hold. And so during periods of stress, trees tend not to use water-based exudates. And now exudates are water-based, so things like resins and kinos, latexes, those things that often produce far less exudates. They're produced by specialized cells in the plant. So they just slow down and stop. And, after wet periods, they'll increase. But one of the things that you'll notice, the holes of the weevil are round. Most weevils make round holes. And they pupate in the zone between the outer bark, the phylloderm, and the xylem. And they'll have lots of stringy bits of wood. So they'll tear bits of wood to make their pupal chamber. And we'll see some photos of those over time. So yeah, Cupressus is a problem. The bigger problem with cypress is having sufficient resources. And in particular, we like to plant them in hedges. They're sort of with a fad planting of about 30 years ago, and people would put them in hedges. And I liken it to starting a farm in a quarter-acre block of land, and that's fine. You look down at your cute little rabbits, and you think, yeah, farming rabbits is good. And as you come back a few months later, you realize your rabbits are actually baby sheep. And now you've got 20 baby sheep in your quarter-acre block of land. It's getting to the stage where it's challenging until you realize that you misidentify them and they're actually calves. And that's okay. And go along another 6 months and you realize that the calves are actually small bull elephants. And you realize that there's no way the land can sustain it. And that's a little bit what planting a hedge is like. You get to the stage that as the hedge gets bigger and bigger, the resources that are available are more and more challenged. So the recommendation is if you've got a hedge of cypress, you want to hedge them. And we see them planted, and I'm assuming all of you do. Just give me a thumbs up if you see them planted at half-metre intervals even. And these are trees that'll grow to 70 metres high. That'll grow to a canopy spread of, 20 or so metres. That's not something that you want to be planting at those spacing unless you're going to hedge it regularly. So you do need to hedge them. So anything else you want to add on that, Vicky? Just make sure that we've covered things.
No, I think what we were going to do was take out all the dead parts and try and save the other ones either side.
Yeah, look, I would run in a wetting agent to make sure you've got moisture going in well, and some supplementary irrigation. If there's adequate water, water's the #1 shortfall that normally always affects plants in Australia. Not all the time, and sometimes at the moment it's the opposite. If you're up near where Peter is, it's too much water, and I'm starting to see trees dying from anoxia. I saw a grove the other day. They filled around with clay, and we'll talk about that at some stage or other, that caused anoxia to occur.
This photograph was very recent. Do you think it could have been too much water?
Is this Sydney based?
Yeah, Sydney based.
What suburb roughly?
Engadine, southern Sydney.
Yeah.
Oh no, out past Engadine, yeah, further out.
The sandstone based soils tend to drain a lot better than heavier clay soils. Most of the ones that I've seen have troubles recently have been where people have brought in clay and put clay over the top of another soil, and you only need a small interface, and that clay becomes saturated. It won't let out. Clay water is very clingy compound. It likes to hold on to things, and the smaller the sort of soil particles, the more it'll cling on in to bits. If you go back to my earlier days when I was just a young kid, my mother would go to the nurseries and we'd buy plants in terracotta pots. Terracotta pots made out of clay, and they wicked the water out. And so they used soil back in those days. The problem became when we started to put soil in plastic pots, and the plastic pots wouldn't wick anything away, and your bottom of your pot will fill up with water. And the site that I went to the other day that did have real bad anoxia on a grove of trees, the filled material was absolutely saturated. I sank down to my knees, whereas the area of natural soil, I sank down to my ankles. And that's just the difference in how the water was penetrating and getting away. It was only affecting the surface when it was natural soils. They hadn't been altered, where the introduced soils had been put in that hit the original soils, the interface zone, and then that water would not soak out of there. It stays in there and just builds up until it gets completely saturated.
The neighbouring property had done some retaining with timber garden beds in recent times.
And so they may have done some root cutting.
Yeah.
And as I said, the big challenge always for most of our trees is water. One of the things that's a problem with cypress is that they don't have, and a lot of the gymnosperms, is they don't have big leaves. And so you don't notice things like wilting until it's way, way too late.
Yeah.
You'll often look at a row and you'll just see the early starts of the changing color, and by then it's usually too late. NASA invented a pair, or developed a pair of glasses for you to see plant stress earlier. Google's been playing with it of late again, looking at spectra to deal with it. They give everything a purpley hue. Don't drive with them on, it makes you feel like throwing up. But a trained eye will pick the couple of shades difference fairly quickly. The NASA glasses just make everything look a lot more obvious. It makes the shift to the yellows more conspicuous.
Okay, thanks.
No worries. This doesn't look so pretty, Peter. This one we're going to talk about tonight. We'll go right up nice big if we can. Okay, some fruiting bodies. This is a spore print and really important for us diagnostically. And the spores are white to creamy white. And there's some inverted V's. And this is what I was saying about that earlier image on what I said was Genopolis. There's some damage going on, these fungal lesions. So this is almost certainly Armillaria. And I suspect Armillaria ludobovalina, again, from the fact that it's been done in Victoria. And this is a primary pathogen. And we'll talk about primary pathogens. There's a few we deal with. But primary pathogen says, I don't care how healthy you are, you're going to die.
Mark?
Yep.
Do all armillarias have the characteristics ropes of tissue that goes up the trunk under the bark? What is it called?
Fungus, which is this one, Armillaria ludobovalina, is a root-borne pathogen. That is, it's stuck to the root. It can do perhaps a few cells past the root, but it in reality requires root-to-root contact to spread. And we'll talk about it a fair bit more tonight. But the important thing is to remember it's a primary pathogen. Thank goodness it's not really good at reproducing. And there are things we can do to stop reproduction. There's things that we can do to stop the virulence of it in the built environment. So there are some things that we can do, but yes, that's a nasty one. Great image. David, would you mind sending those images through in a higher resolution at some stage? David's, would they?
Yep, no problem, will do. Just one interesting thing with that fungi, I worked for some Italian people for a long time, and every winter when that would start to develop, they would go crazy and go and collect it and eat it by the bucketful.
It tastes great. It's very hot and spicy, so it's like cooking with chili, but it doesn't have the same chili flavour. So it's got that hot peppery chili thing to it. And yes, armillarias are edible. So that's a thing. Got to watch it because there's a very similar fungus called Amanita phellus, and it's the death cap fungi. And usually 3 to 5 of those is sufficient to kill an adult. Always exercise caution if you're eating fungi, but once you know what it is, it certainly is edible. And whilst we're tasting fungi, anyone can eat a pea-sized piece of fungi, chew it, spit it out. It's never going to kill you. I always recommend spitting it out. Some of them, can be a little bit unsettling on you. And my daughter is a foodie, tried her first armillaria and said, yeah, I can see it's hot, and then swallowed it and said, oh my, it is hot. So I can tell you it's hot as it goes down when you eat it raw. Michele sent these creatures in. These are larvae, and they're sawfly larvae. They're the larvae of steel blue sawfly, Pergera, and it's important to remember that the adult for this is a orangey and blacky brown colored wasp. It is not a blue wasp. I don't know where it gets the name steel blue sawfly, because the larvae is not steel blue, and there is a flower wasp called the blue flower wasp. Let me show you a picture of it. That you don't want to get confused. The blue flower wasp is a desirable wasp that attacks scarab beetle larvae. So don't go getting those two things confused. Blue flower wasp. So this is a blue flower wasp. You'll see it around, flying around mulch areas in particular. Striking looking wasp. It lays a single egg inside the scarab beetle larvae, and it's a parasitoid. Parasitoids are parasites that kill their host. So this will eventually kill the scarab beetle larvae. So it's desirable. On the other hand, if we look at steel blue sawfly, and you'll see why common names are a real problem when it comes to lots of things. this is the adult steel blue sawfly. So nothing to do with steel blue. The larvae doesn't look steel blue, at least I don't think it does. And certainly the adult does not look steel blue, but it's a sawfly larva. They're gregarious, they're predominantly nocturnal. They use the eucalyptus oil to defend themselves. And I don't know whether we talked about this before, and I'll probably get into trouble, and I'm good at getting into trouble. Politically correctness is not my best forte, but if you want to feel some fairly good physical discomfort, pour 50 mils or 20 mils of eucalyptus oil into a bath and have a bath. You'll find parts of your body that'll burn like you've never believed things could burn. And so these guys get this horrible reputation of being called spitfires. They don't, when they're disturbed, they simply regurgitate back up eucalyptus oil and use that against things. And eucalyptus oil is a really good chemical for doing lots of things. It's actually a reasonably good insecticide. It's a good antibiotic, except that it burns. These guys aren't spitting that out as such. They're just regurgitating it up and making themselves unpleasant. Overall, if I see them, I'll just remove the small little section that's got them, but they seldom get to the stage they get out of control on the trees. And guys, I think that's it for the, we covered that one before, so great. Let's now have a look at what we're doing tonight, which is fungi. And you'll have to excuse me whilst I open up the PowerPoint. If it'll let me, it will. Maybe it won't. I'll go for Arborist PowerPoint. There it is. And slideshow from the beginning. And I'll just close that down if that's okay, guys. I apologize for that. If I don't see something. So fungi, when I was a young kid, it was a part of the plant kingdom. In fact, it was right up through university. There was a proposal to move fungi out of the plant kingdom as early as the late 1960s, but things are slow to change, and certainly general science is very slow change. So let me see if I can just make that a little higher so it doesn't cover up any of the script. Okay. So what are fungi? Well, yeah, they're different than plants. One of the first differences they've got is all cells that are complex, what we call eukaryotic cells, have membranes. Lots of things in membranes as well. Membranes are just a chemical that makes a container that holds things, something similar to a bubble. You know, the bubble holds in air. Well, we've got these chemical things that hold in a whole pile of different bits and pieces inside. And in a plant cell, we've got the cell membrane, and then we've got the vacuole membrane called a tonoplast. We've got organelles in membranes. We've got the nucleus in the membrane. So this membranes are a really good chemical holding thing, and it's made out of phospholipids, whatever they are. And they do a great job of holding things. But plant cells also have another container on the outside, and it's made out of cellulose. And the easiest way I can describe cellulose is cotton wool. So just think of it as having this canvas bag on the outside. And that helps contain that phospholipid membrane. On a human being, we've got a skeleton on the inside that gives us some support. And we've got all these cells attached around it, and that's the case for all things with skeletons or all advanced animals. They've got the skeleton that exists. And then on the outside of a human being, we've got this area called the epidermis, or upon the skin. And these are just dead cells. And you know that they exist when you shower, particularly if you've got a long, hot shower and you scratch yourself with your fingernails, you'll see big piles of stuff come off. They're dead cells. So we don't have this fancy outside container. Well, plant cells do, and so do fungi. It's just the difference is, whilst plants are made up of cellulose cell walls, fungi have a chitin cellulose cell wall. And chitin is the chemical that is used in the exoskeleton of things like insects. So a lot of our insects, that brownie color exoskeleton, think of a cockroach, the outside crusty shell, that's made up of chitin as well. They're heterotrophs. Hetero means other. Troph means to feed. They get their food from other sources. Whereas plants are autotrophs. Now, like everything in nature, when you start making generalization, nature mocks at you. There are a few plants that don't produce their own food. And I'm not talking about mistletoe. That produces its own food. It just gets its water and solutes from the host plant. But there are things like Thesmia and Indian pipes. Thesmia is Australian, Indian pipes American, that are true parasitic plants. That is, they get all their food from other plants. So fungi are heterotrophs. They get their food from other sources, and that's important. They can be single-celled. This one's yeast. And I know that a lot of arborists like yeast because it does wonderful things like produce alcohol. It also does things like make bread nice and light. They can be multi-celled. This is a turkey tail fungus. We've got lots and lots of these fungi that form different structures. And it's important to understand that our knowledge of fungi is fairly early. We're really good at the big guys, these multicellular ones, but the things that don't produce big fruiting bodies, we're not so good at. And so we're somewhere, it's estimated, about 5% of our fungi species have been named. So there's a hell of a lot there that we still don't know and understand. And every now and again, one of those comes to the fore in arboriculture around the world and becomes a problem for us. They can reproduce sexually using spores. This long bag here is called an ascus. And so this is an ascomycete. That's one group of fungi, and they produce 8 spores in a long tube. And that's one of the ways we identify them. This is a shot taken at about 1,000 times magnification. So it gives you an idea on how small these things are. Not big at all. But they can also reproduce asexually, vegetatively. And that's another way we spread things around. So the hyphae can be spread around. They can also produce a number of really odd, I'm going to try moving that to the bottom. Number of different asexual spores, and that's different than the way we see the world. These are like seeds that are clonal seeds. And here's, for example, the teleospores of the apple cedar rust. We don't have in Australia, it's a shame. We do have a whole pot of rust, but this is quite a neat little one. Teleospores are overwinter well, they're dark coloured, and dark coloured spores have some advantage over light coloured spores. And we'll talk about that when we go back to the Australian honey fungus. Things can be also quite microscopic. This is aspergillus, can cause health problems for people. And it's, again, quite pretty, but microscopic. That's a scanning electron microscope and micrograph, sorry, and that's total image is about 0.4 of a millimeter across, just over 0.4 of a millimeter across, so quite small. So they can be very large. This is a bolete. A bolete is a fungus that looks like an ordinary mushroom type thing, except instead of having gills, they've got lots of little holes. Okay. Those ones that produce gills, sometimes we call it garrix. It doesn't really matter. They're fungi with big fruiting body. And they can be microscopic. Here we've got a Sporodochium, and these are producing asexual spores, conidia spores, just huge amounts. And this is a 1-year-old twig, and this is the bark on it. So you can see tiny in terms of scale. And this is an image by George Barron, by the way. George was wonderful enough to make his images available of all his work to me, which was fantastic. So most fungi are filamentous. So we've talked about single-celled ones. Then there's these ones that want to grow these strands of hyphae. And they can branch, they can grow longer, they can put a new one at the end, they can divide, but they can never divide sideways. So they're not like trees where they can get thicker. Again, one of those differences between fungi and plants. And when they come together, we call them mycelium. And so here you see the mycelium. And I guess if we had a really good shot with a magnification on this, you'd be actually able to see the hyphae individually there. So again, young man at university, fungi are part of the plant kingdom. By the time I got to looking at fungi as an arborist, we started to really say, no, look, fungi are in their own kingdom. And then there are things that we thought were fungi, Phytophthora cinnamomi, or the whole Phytophthora group, the water molds, saying, no, they're not fungi anymore. And there's good reasons for. First of it is their cell wall is made out of cellulose. And I said, that's what plants have. So cellulose cell wall, not a chitin cell wall. Second thing is they've got a reproductive spore called a zoospore that has flagella. They're like little motors, and they can swim, just like you see seamen, for example, swimming. These guys can swim and move around, and so they move through water. And this really gives them some advantage. They like wet conditions. And Vicky, you were asking earlier on about, could the current rain have an influence? Certainly the rain does. After we've had a drought and then you get rain, you get a stressed tree, you get some stressed roots, some roots that are damaged or dead. And these guys are very, very weak pathogens. They're not strong. Some plants, they're really good at knocking around, and the typical one for these are things like Proteaceae and grass trees or xanthoreas tend to be very susceptible. So if you're seeing a grass tree, those of you in Melbourne can go down and look at Flagstaff Garden, I think it is, has got some up the top end, grass trees that have been planted that are probably dead by now. And you can see the damage coming in from the Phytophthora. The things you can do to control Phytophthora, there are a number of chemicals that you can use to treat Phytophthora. So there are fungi that affect, chemicals that affect the fungus directly, and there are chemicals that stimulate the plant's natural defense system. And so both those exist. The latter group were developed in Australia to help manage Phytophthora in Australia, and we introduced it in Australia. In fact, we go back historically, many of our plants were moved around in the 17 and 1800s as whole plants because we weren't so good at figuring out how to propagate. And so digging out things, cutting them back and moving them was commonplace. And so we introduced Phytophthora to Australia, but had we not introduced it back in those days, we would have anyhow, because these are often spread around by things like shoes and golf shoes, anything that gets a little bit of dirt on, you pick it up and you can move it around. Phytophthora is not good socially with fungi. So one of the ways you can really knock Phytophthora back is to make sure you've got good mycorrhizal associations and good mulch layers. So they're weaklings. They pick on stress plants when there's no one else around to attack, but otherwise they're not particularly good. So slime molds are the next group. These are much the same, and I encourage you to go and have a look at the videos on slime mold mapping out areas. They're really quite clever. The reason these aren't considered to be fungi is that they move. All of these hyphae here were once spread right throughout the mulch, and they've come together over a few days, and they've actually moved around. So they're capable of moving around. So again, they're not in the fungi group. They're in that group that's closely related, not in the same order, not the same kingdom. So slime molds are almost never a problem. They're almost always beneficial, unless you've got something that's in the very final stages of its life, and then everything's open to attack at that point of time. So I've only ever seen it attack a tree once, and that was incredibly stressed and in its last stages. Someone's asked a question. Let me see. Yeah, fungal sporing body, sporocarp. David, is a more technical name. If you're talking about basidiomycetes, then we technically call them a basidiocarp. And if you're talking about ascomycetes, we call them an ascocarp. Yeah, they're not fruiting in the sense that plants do. And I guess this is a bit of that heritage of things changing. So remember I said when I was at university, these were in fact still a part of the plant kingdom, although the process to move them across had started. And again, if I go back to when I did some further studies in mycology, they were still in the fungus kingdom at that stage. So it did take time for things to move out. So yes, indeed, you're right. Sporocarp is the correct technical term. These slime molds, my typical joke is that they can plan and do a networking of a city or a rail link or a road transport system very effectively. And I figure that's funny because town planners have troubles doing that work, and these are primordial slime, so you can figure the rest out. So fungi are mostly good guys. We said we don't know a whole pile of them, but most of the fungi that we deal with are beneficial. And this is one that actually got me into contact with George Barrett in the first place. This is a oyster fungi, and it's a fungi. The strands here are the fungus. This is the hyphae. These are a nematode feeding fungi. So they like to feed on nematodes or small little microscopic worms. And so they capture and restrain and kill and then consume the fungi, the nematode. And in all of these pictures, you'll notice there's no mouth on the fungus. How they consume things is they exude a thing called an enzyme. The enzyme breaks things down, and then they suck that material back in through their cell wall and utilize it. And I liken it to something like a bird spider. A bird spider, an Australian spider, does much the same thing. Those fangs are just to put a venom in. It's not to eat them, and it's to put an enzyme in. The enzyme will dissolve a small mouse or a bird in a matter of a day, just turn it to pulp, slosh. And so enzymes are used by fungi to break down things. And that's important for us later on. And again, George Barron's image. There are fungi that eat fungi. So this is a fungus feeding on a fungus. And there's a whole pile of mycophagous fungi. One of the better ones that we see played with is trichoderma. Craig Hallam's been doing some wonderful work with Francis Schwarzer on trichoderma and the control of one of our primary fungus that we'll look at in a few more minutes, Phellinus noxious. This is a good fungus. This is a mycorrhizal fungi. This is an ectomycorrhizal fungi growing on a oak root from the University of Sydney. And you can pick that this is mycorrhizal. There's a big layer on the outside called a Hartig net, and the roots have stopped growing longer. And what's happened is there's been a little chemical dance going on. The spore of the fungi germinates, it starts to grow towards a root. The root then reacts, it produces chemical defenses. We don't see these defenses because they're chemical and not physical, but it'll start to produce a response to say, hey, what are you doing here? And the fungus says, look, it's okay. I'm not a bad guy. It's me, Micorrhizal. And like all good Irish party people, the root and the fungi get along, and the fungus takes over doing some of the work of the root. It takes up water, it takes up solutes.
Mark, what sort of root is that? Is that an absorbing root, or is it a different part of the root
okay, so this is a young pioneer root that then there's root hairs are highly reduced. So when you get a mycorrhizal association, the root hairs almost don't extend at all. They're just slight little bumps. And the fungus does the rest. The fungus forms around. So 2/3 of that root at least is mycorrhizae on the outside, hyphal strands.
These are relatively ephemeral.
These are fairly long-lived, Peter, oftentimes more than a year, sometimes several years, very efficient. And very efficient because surface area of fungi is way, way higher than the surface area of an absorbing root. Significantly higher than the surface area of a root hair. So these guys have got it over plants and being able to take up things. Not only that, but remember I said they produce enzymes? Well, one of the enzymes they produce can take phosphorus. And phosphorus is bound, it won't move. And it can break it down to form a phosphate. And phosphates are movable. And if you want to see some really good things, there's some on, you'll find it on Apple University. You'll probably find it on a Google search. The Open University has done some wonderful demonstrations of this relationship between fungi and phosphorus and being able to make phosphorus available to plants. We know it in Australia. Peter, when you and I were young, we had a thing called the superphosphate levy. My grandfather was somewhat involved in that process as a wheat geneticist and wheat breeder for the Department of Agriculture. And we all know that you add phosphates in, plants grow better. Well, what we didn't realize is that as we're adding phosphates in, we're damaging the mycorrhizal fungi. So mycorrhizal fungi are extremely sensitive. So you put high amounts of phosphates that don't regulate phosphate uptake well, and it kills them. And we were doing that with our wheat farming right up until about 30 odd years ago, I guess it was, maybe a tad longer. The first process of stopping using phosphates was as a drop in production, but over time the production increased, and that's because there are, again, mycorrhizal fungi in wheat, glomulaceae, vesicular muscular mycorrhizae that grow in the inside and into the root, into the cell wall, and membrane touches membrane. So these fungi, really important, but were damaged by the use of phosphates. And we also, of course, turned our streams green with algae, made algae grow like crazy because the phosphate's highly mobile, it washes away very quickly, which is why we had to keep on putting on phosphates after phosphates. It's not because the plants used it all, because a large proportion would wash away. Apparently, if you just let it go, the fungus will do its job. And if you want to have a discussion, Adam Tom's family's got a reasonable wheat production. Craig Hallam's family the same. You can always have a chinwag with them about the change in farming practices, how our model of a plant in a laboratory is different than a plant in a natural environment, and that's because they are in complex associations. How do you feed the beneficial fungi? Well, when it comes to mycorrhizae, the answer is don't use high phosphate fertilizers. That sort of means the native stuff. And the reason the native fertilizers work well is that a lot of our native plants, particularly our proteaceae, have proteoid roots, and proteaceae don't regulate the uptake of phosphates. Well, you might have heard me say that earlier on about Micorrhizal. And same thing, you add too much phosphate in for proteaceae, it kills them. So avoiding the use of
What about poultry wood chip mulch?
poultry mulch is reasonably high in
Whole tree.
whole
wood-
tree mulch. Whole tree mulch is great, but it doesn't feed Micorrhizae.
Okay.
Micorrhizae are fed by the tree exclusively. It's a what we call a mutualistic symbiosis. That is, it's beneficial to both, and it's both are being supported by the process. And some trees, Pinus is definitely one. Pinus is an obligatory symbiote. It doesn't grow well at all, just struggles and goes nowhere if you don't have a mycorrhizal association. And to deal with that, we actually brought in mycorrhizae. You've all probably seen the orange fungus that grows underneath pine trees with a little nice, or cedars, gymnosperms, with a nice little orange and white things that form a fairy ring. That's Amanita muscaria, and that is a mycorrhizal fungi. So Amanita is a mycorrhizal fungi, and we introduced that Amanita Phellinus to Australia, probably indirectly, but certainly intentionally we introduced muscaria, Amanita muscaria to Australia. But what you can do is make the environment conducive for the fungus to grow. So ectomycorrhizae fungi, these ones are often grow on from puffballs, but as I've said, not always. Amanita certainly isn't that thing. If I plant a new Pinus, I like to do so in autumn, and I like to go grab a fruiting body, a sporocarp of Amanita muscaria, and put it down on top of the pot and make sure there is a positive infection, that the spores drop. Mulch is good because spores that drop during the night, and most of these fungi, the spread daytime, 15 minutes exposure to UV light and you've burnt to death. You're not going to survive. So night spreading of spore is very important. You want it to land, and then you want it to get in out of the light. So mulch is good for that. If you've just got a hard surface like, compacted soil, it's going to sit on top, the sunlight's going to hit it, and it's going to be gone. The spores of these ectomycorrhizae are very small, very easily distributed. The other ones, things like wheat, the Glomulaceae group, the vesicular buscule mycorrhizae, or what we used to call, if you go back to some of the older books, you'll see them called endomycorrhizae. They're big and they're not easily moved through the soil. So organisms are important. They're things like wood roaches and worms and cultivators that move things around, move soil. And so what you can do to encourage those, again, is don't scrape soil back. Fortunately, they form an association with turf grass. So turf grass, when it used to have roots on it, used to bring some in. You get almost no roots on turf grass now, but you do get some come in. And that's probably the only good thing in my view about turf grass. But nursery logs is one of those things that really is important. You get a whole pile of different organisms associated with bigger logs left in your mulch. So, try and leave some 30 centimeter diameter bits of wood with your mulch, and that'll make a huge difference. So hopefully that answers that question for you, Matt. Okay. That's so good guys. Lots of good associations there. Bad guys. This is Armillaria in its very early stages, Armillaria ludobobolina. And it's a primary pathogen, as we've said. It's easiest spread around. It's got a white spore. White spore means no melanin. Melanin is that lucky thing that 3 out of my 4 children, and my wife's just over a half caste, just under a half caste, Wiradjuri woman, and 3 of my kids have got dark skin. They get in the sun, they go brown. I've got freckles, so I've got lots of spots that don't go brown. And that melanin's got a protective quality against UV. And same with these spores, that dark coloring gives protection against UV damage. These are white, and we saw from David's photo earlier on, and from this one here, this one's an image by Danielle Austin. This is white to creamy colored spores. So this is Armillaria. That's taken from one of these as it opened up. And they are often associated with trees that have recently been cut down or have recently snapped close to the ground, and the fungus gets in and takes over. So the big mistake here, tree was cut down, stump was left, spores got in, managed to survive, so spread at night during rainy weather, got into a little crevice somewhere, the spore got to a spot where it wasn't having problems. And this is a pathogen. So a pathogen is something that likes to kill living cells. So it wants something that's only freshly dead because there's still living cells in a tree that's been cut down, a tree that's snapped. And those living cells can survive, by the way, for a number of years. You know, there are records of them surviving for a decade or more. And that's because they're parenchyma, and some parenchyma is long-lived, or parenchyma, depending on what part of the world you're from.
Can it also act as a saprophyte?
It acts, it's a shocking saprophyte, it's lousy. And so that's why it wants to get going in something that's dying dead just recently. So cut down. So one of the things you can do is get into the habit of cutting stumps closer, covering them with mulch, or cutting them a lot longer so that the fungus doesn't really get a chance to get in with before other fungi come in, or grinding the stumps. And that'll help the spread of fungus. Once it's in, it's root to root contact. So this site had an avocado in the backyard. My guesstimate was it's just a question of time before the avocado gets infected. And it's just a matter of time before every tree in the surrounding block gets damaged. If you've got a nice big wide road, often roads will stop it, but they spread under roads when you get root to root contact, and they will take out any tree they infect. But
Mark,
yes,
do trees have root-to-root contacts, even completely different species? Like, you know, this could have been, what is it, maybe a pear tree or a rubinia to an avocado or an eucalyptus, or because if they're just touching, the fungi shouldn't be able to jump, as there's the cortex protecting the bark, or am I thinking it wrong?
So if the root's touching another root, let's have this, and I'm going to guess, say prunus, I think it was, looks like prunus. If this prunus root is growing past and comes into physical contact with the root of the avocado, that's not a problem at this stage. The problem is, as the Armillaria gets going, it gets momentum, it gets energy, and its ability to attack, its army, so to speak, of cells increases. And so it's able to break past the defense systems of healthy trees and completely kill them. So it doesn't need a graft, it just needs contact. And there's a possibility that it can extend a few cells long, but when you see how long a cell is, it's, you know, it's not significant. They're not able to travel any distance through the soil, unlike the American Armillaria melia, or Europe, it's in Europe as well, I believe. They produce these big long strands called rhizomorphs, and we don't have that on this species. This is purely root to root contact.
Is this a native fungus?
It's native to Australia, yeah. And it's a big problem, because you've got to think about how often this is going to colonize. So I can think of 8 or 9 colonies in Sydney. I'm sure we've got every arborist together. We might come up with 50 or 60. You know, that's 60 street blocks in a a city the size of Sydney, that's not huge. But they do. I mean, I can think of a colony down south at the moment that's going neighbour to neighbour to neighbour. I can think of one of the eastern suburbs that's doing exactly the same. This one is in Narrabeen, and it's doing the same. It'll eventually spread right through the area until you get a gap or a separation of root-to-root contact. There was an attempt, and it worked reasonably well for quite some time, at the National Botanic Gardens in Canberra to isolate Armillaria that they have there using a physical barrier. The physical barrier they used was an emergent herbicide called Casron G, and Casron G is a contact herbicide that when you come to contact, tissue dies off, so they use them in nursery, stop young weeds coming up around nursery plants. Doesn't hurt unless you come into contact. In this case, the barrier was set in by digging a trench and filling it with a mix of Casron G and media, and roots couldn't penetrate through that. Finally, they did breach, and the fungus is now breached. So I did a paper for it, and if you want to ask Peter, and he'll put it on the Dropbox, that talks about some of the work that is being done on control, but we haven't had much success to date in controlling it. Been quite poor. There are a few other species of Armillaria in Australia, and none of them are a significant problem, just this one species. So white spores, that's a good way. And I said a little bit of the sporocarp or the cap, take it, chew it. It'll be quite peppery, quite hot. And that's not always great when it's not an open sporocarp, because a number of unopened sporocarps are hot and spicy. So you really need an open one to get to the stage that you test. A spore print works well. The problem with spore prints is that you need to know some idea of what you expect as an end result. Because we're pre-identified with taste and the physical features, my suggestion to Danny was she just use black paper, which she's done. But if you're not sure, black and white paper, and the way you get black and white paper, half black, half white, is to get a piece of black paper or black anything and photocopy half. So you've got, you put your sporocarp on one half, and that way you can see if it's a white or a coloured spore. So this is just a mass of spores that's been left behind. And David did one. Genopolis, which is the other one we looked at this morning, there's earlier, commonly mistaken for it. It's not quite the same. It looks different, different telltales, but the best telltale is underneath. If you remove it, they have an orange spore, and you can see here there's distinct orange spore that's visible without anything even being moved. They do produce large amounts of this spore that's quite orangey in colour. And these guys, again, are reported occasionally of being very weak pathogens. We talk about very weak pathogens. They simply are the first to get in when something's on its way out and dying. Phellinus noxious. This is a bit more of a concern, a lot more of a concern. Phellinus noxious is called brown root rot. That's its common name. And that was a common name given to it by a scientist who was a moron, because now we get it commonly confused as being a brown rot. It's not. Francis Schwartz has done the analysis of the enzymes, and it's clearly a white rot. But in the process of decaying roots, it does discolor them, turns them a browny color, and hence the common name brown root rot. It would have been a lot better to have called it Phellinus root rot or something like that. We didn't. And it can produce a crustiness close to the ground that you can see. So this is a little bit of crustiness here. The next one is from Lismore, and this one failed. And you can see the crustiness has gone up the stem quite an amount. It's a little bit like, and I say this every time, it's like Kretschmeria. For those of you European Micheli, you understand this. Thank goodness someone does. It's a little bit like Kretschmeria in appearance, but it is definitely Phellinus.
There are some subtle differences between it and Kretzmeria, I believe.
Yes, fair differences, but it's that likeness. It's like saying casuarinas look like conifers. They're clearly not, but they do share some similarities. A couple of questions have come up. Over the last couple of years, the gardens here in Canberra have had Armillaria spread. Oh yeah, it's broken the barrier. And yes, quite a number of trees. It's a primary pathogen. I was doing a court case last year, and the barrister was alluding to the fact that, you know, having a tree that's stressed makes it more prone to attack from Armillaria. And trying to explain it to him, the first thing that came to my mind, it makes me laugh now, a primary pathogen is like me sitting with a 357 loaded, it's a pistol, a handgun, loaded and pointed at someone's temple. It doesn't matter if they've got cancer and they're dying. It doesn't matter if they're in the prime of their health. The moment I pull that trigger, you're dead. And that's what a primary pathogen, it doesn't care about your health. Whereas secondary pathogens do care. And so there are differences there. And we'll talk about a couple of those secondary pathogens. So the dilemma with this is it's a tropical disease that is moving south. And we're not sure on how far it's going to end. Craig Hallam's suggestion was around about, isn't it horrible?
Port Macquarie.
Macquarie, thanks, Peter. Port Macquarie.
I can see no reason temperature-wise why it wouldn't be down that far.
I think it'll go down that far, and I suspect it'll go all the way down to Sydney, to the coastal regions. The reason for that is there have been a number of secondary disease or other diseases that are tropical, like lesser coconut weevil now that's in Sydney, and I'd be surprised if lesser coconut weevil hasn't hit the coastal areas of Melbourne. You only see it on the coast, you know, within a kilometre or two, it starts to reduce in occurrence. So I suspect.
What's the temperature range for the Phellinus noxious for it to be able to survive and reproduce?
Not sure that.
It likes to get going at about 18 degrees. It doesn't really do much. It sits pretty still below 18 degrees. And I don't think it likes temperatures below 8 or 9 degrees much at all. And so those cold temperatures seem to restrict its spread.
But now keep in mind that soil temperatures can be quite different than ambient temperatures. So
that's right.
These are soil temperatures.
I suspect we'll find it as a metropolitan disease in Sydney and eventually in Melbourne along the coastal areas, and certainly, you know, it was reported down to in 2014. Within 4 or 5 years, it was found in Grafton, and it's heading further south. So it will go further south. I'm pretty sure of that.
So I'm not sure that the reason that we don't know it in Coffs Harbour and Port Macquarie right now is that it's not there, or it's just that it hasn't been noticed.
I think that's a fair comment, Peter. And I think that, you know, it's just that process. And I know that there's been several failures now in Grafton. So I suspect that it will move and continue to move. But again, this is something we don't know. It's just something to keep an eye out. Be aware that it's there. Peter, were you in the class when we found it in Grafton? Was that your cohort?
No, it wasn't, but I have had dealings with infections in Grafton.
Yeah, so I was down in Grafton with a class from Wallingbar TAFE, and looking at trees, and went past 2 Ficus microcarpa helii and said, oh my, that's Phellinus noxious. It was over a fence, we could see it, and we went in and looked at it, and sure enough, one of those trees failed several years later. Okay.
Is it an Australian fungi? Sorry, Mark.
It's
going back to that
Australian.
Phellinus.
So it is not just in Australia, it is through Asia as well. So it's quite a broad spread fungi.
It's a big problem of fruit trees in Taiwan and places like that. Taiwan is where they did a lot of the first work studying it.
And
So
Peter,
this is
passion
kind
for,
of a new pathogen then, because if you talk about it's moving, it hasn't reached its full area of
Yeah, well,
its
see, we
full area,
suspect that
potential
we-
area.
we suspect that it will move, especially with global warming as the temperatures increase. But just because that we don't know that it's in Coffs Harbour doesn't mean that it's not there. It just means that we haven't noticed it. And
Okay.
I know from myself, when I used to go to conferences in Brisbane, the people would be talking about this Phellinus noxious stuff, and I would just shrug and go, "Oh, yeah, nothing to do with me." But that was only just because I hadn't noticed it. And in fact, when I did notice it, I started noticing it everywhere.
yeah, it's certainly. I know, Peter, that when we're up there, we found it in the middle of a national park.
Yeah.
We found it all over the place. But I suspect that hadn't been there that long. And I think the same thing happened with Brisbane. It was, something that wasn't a big issue at all. And then I think it was a Newmarket in.
Yeah, the fig trees started to fall apart.
Yeah, a number of trees and jacarandas. Because it is a pathogen, let me give you a general thought, and it may not be accurate all the time, but pathogens like living cells, they kill living things. That's their real kick out of life. And so trees with large amounts of parenchyma are often more vulnerable. Ficus has huge amounts of parenchyma. Jacarandas have a high amount of parenchyma. Ukes don't have as much, and ukes aren't as exposed, but they do attack ukes and uke plantations.
But having said that, Mark, it's been recorded in 200 different.
Oh yeah, it's polyphagus, so it doesn't care about its.
Yeah.
But
I
those,
think I've seen it in Camphaloral.
yes, and a Camphaloria again has got lots of parenchyma again. So things that are high in parenchyma are going to favour its mode of attack. That is it's not that it's going out and saying, wow, I look at this as a tree with lots of living cells. It's that once it gets in, it really has an advantage in that environment. Whereas, something that only produces one layer of living cells, some of the oaks, for example, may have a lot greater problem in getting going. And time will tell. Okay, so then the other reason we have concerns about fungi is decay. And we have two sorts of decay that we generally are concerned about in trees. Every now and again, we throw in a couple of weird and wonderful secondary groups, but they're fungi of mostly lumber or timber. It's not that they can't affect trees, it's just not the primary issue. So the first one is brown rots, the second one's white rots. Brown rots take away the cellulose. I said cellulose goes around the plant cell. So they have an enzyme that breaks down cellulose and they leave the lignin. Brown rots, by the way, you can see this is a gymnosperm. Those of you who are really good with your anatomy, you can see uniseriate rays taken at 10 times magnification. And this is often Armillaria. There's a white rot down the bottom here. White rot takes away the lignin and just leaves behind cellulose. And you can see lots of hyphae here as well. You can see the mycelium. Can't see the hyphae individually. So white rots and brown rots, and this, interestingly enough, were both from the same host. So fungi can move through a plant cell, and this is a single cell from here to here. These are called septae. Some fungi are septate, some aren't. This is a trachea, so this is a gymnosperm, just like we're talking about, and you can see how easy it is for a thin little tube like a trachea. A trachea is a single cell. This is about a third of a single cell here. from one side to the other. You can see how much smaller fungi, so they can move through the tree fairly effectively. And this is where we get compartmentalization from. When we talk about the walls, there aren't physical walls. It's just the way in which it's easiest for fungi to work. And there's a whole concept on this out of Germany called compartmentalization of dysfunction of decay. It's the same thing. It says that fungi can't move in cells until this environment becomes conducive for fungi. Fungi need air, they need oxygen. And so they require an injury. And there's wonderful work by Professor Limbodi on a lot of our fungi in trees starting as endophytes. That is, they're existing inside the cell, inside the tree whilst it's living, and they're just there as a single cell. An Australian mycologist, Dr. Peter McGee at Sydney University, again did similar stuff. He played around with leaves. He sterilized the leaves, put them in an agar solution, and fungus started to grow. And again, they're just saprophytic fungi, but they're inside the leaf. They're ready and going. And these fungi can be beneficial during that time. They can actually help the plant's natural defense system. So Matt made a comment, definitely seen Phellinus noxious newly trained aerial roots, ate them up, let alone the trunk. This is in Queensland, hopefully, Matt, not somewhere south. You might let us know if it's somewhere south of Grafton. Grafton's as low as we have it formally recorded. So yes, fungi moves through the transport system. Matt's just given me a response. Brisbane, yeah. Yeah. Parenchymus high in ficus. It's just a prime candidate. You can see here that to get to the next cell, and by the way, this is blue stain fungus on Pinus. This is not a particularly problematic fungi, but they've grown through the little connections between the cells. These are called bordered pits. It's gone through and into the next cell. It hasn't actually gone through the whole cell wall. It's looked for the weakest path. And again, this is how we get walls 1, 2, and 3, is understanding this. And if you're dealing with monocots, palms have walls 1 and 3. No wall 4, no wall 2. And we think, oh, they don't have ordered and they don't have compartmentalization. Monocots have wonderful compartmentalization, just only 2 walls rather than 4. So we'll talk about that when we look at infection on one of the palms. So brown rots, they degrade the cellulose and leave us with the lignin. Lignin's like cement. It's not really good until it's got things to bind it together. So we add in concrete, we add in steel mesh to help hold things together. So it's the same. These make trees a little weaker. The good news is, and again, you can see this is a gymnosperm, bordered pits going up through here on a tracheiid. This is scanning micrograph. So the two that we have in Australia, the curry punk, and those were some of those shots that David had provided us earlier on, I believe was curry punk, Phytophthora australiensis, and it's got a wonderful curry smell. They're ephemeral. These come and go. They're not long-lived. The reason we get them to last longer is we put them through a lyophilizer or a big freeze-drying machine. Peter's sent a 1.5 ton machine there that does the job. Once they're dried out, then we've got to spray them with a surface spray because the beetles will still come in and destroy them over time. And you can take...
That's the fruiting body that you're talking about, Mark, not the actual...
Yeah, not the fungus, yes, sorry, the sporocarp. And so this is a problem because the sporocarp disappears. It's only there for potentially a few weeks to a few months of the year, and the rest of the time you don't know it's there. The good news is, as I said, in most eucalypts, this is almost never an issue. Beautiful curry smell to it. I haven't had the courage to cook with it yet, and I'm not saying that it is edible, so please don't go out and kill yourselves. But definitely exquisite curry smell. When I take my samples to workshops, then you can smell them, and it's just luscious. So the other brown rot that we have is Ladioporus portentosus. We talked about this earlier on. This particular specimen weighed about 10 kilograms. It's about 3 days old, and you can see lots of little wounds on it already. These are from insects. The insects are attracted to them in large quantities. In about, well, I've actually got this one preserved. This one now weighs about 400, 500 grams. So you can get an idea of how much water's in them. Sucking that water out is really beneficial because it allows fungus to grow inside. And you can see here, wet spots from the glistening below is coming from the fungus. So the fungus is sucking water out. The sporocarp is of benefit. The insects that bore into these are often responsible for spreading the fungus. A number of them have specialized structures on them called mycangias for spreading fungi. Weevils in particular are really good at spreading fungi to other trees. So again, not very common. You can pick it up with ultrasound. You can pick it up with tomograph. So you can pick it up with a resistograph, and in many cases you can pick it up with sounding. So the only drawback is that in the earlier stages, these brown rots are fairly solid, so the tree doesn't get a message to strengthen up. And so you can get brittle failure. And it's not that it's commonplace, it's just that it can occur. And I'll show you a brittle failure maybe next time round. I want to just get through these, and then so if someone is interested in seeing what a brittle failure looks like, if you've never seen a brown rot failure, email Peter and he'll make sure we show it at the next tips. So our white rots take out lignin and leave the cellulose, and you can see here it's almost like acid being poured on something. It's the enzymes associated with the hyphae dissolving, going through and breaking up. And you know, one of the things that I should point out is if you've seen fungus growing on a piece of bread, you'll see concentric rings. Those concentric rings are, first of all, the metabolites, the things that are breaking down, and then they'll often put out the secondary zone that says, this is my piece of bread, leave it alone. If you come in any closer, I'll kill you. And in fact, there's a secondary metabolite that's pretty important to us as people, and that's penicillin from the penicillin mold. And so these chemicals that fungi and plants use are often a lot more advanced than human beings. And I think I've said that to you earlier in one of the groups, is that plants are chemically rich, organ simple. Fungi sit into that same category, chemically rich. So a lot of chemicals being explored in fungi at the moment for medicinal purposes, for looking at anti-cancer and for all sorts of other things. Very important group. Our most common one we deal with is Phellinus. This is a good Phellinus. This is Phellinus badius or Phellinus robusta. When it comes to identifying fungi now, we used to do it based on morphological features, on things that we could see. And now it's all done by DNA. So this is one of the two. Phellinus sp is what you'll generally put it at. And Carl and Podge tell us that these are basically white pocket rots. And that's important to remember. And again, if you want to know what a white pocket rot looks like on the inside, email Peter, say, can we have some pictures of white pocket rot? And I'll show you. We won't have time tonight. But those white pocket rots actually give some strength still. They're not a complete loss. Eventually, animals will come in. I've got great photographs of sulfur-crested cockatoos doing that, coming in and starting to eat out that material, pull it out and make nests. And, you know, nests are valuable. And in most cases, Phellinus in the stem. So these white stem rots is not an issue. This is one that's happened to the same age. That last one's about 30 years of age. This one's about 30 years of age. This one's growing on the root of a on a Eucalyptus mollicana. And I often get quite teary about this because it started picking the wrong host. And it just was never going to get going or never do anything. And that's because different trees have different ability to resist different fungi. And if we think about Phellinus, the ones that we think are really bad, and some of you from other areas, please turn on your mic and share. But in the Sydney and Melbourne regions, Eucalyptus nicolai and Eucalyptus caparia often lose a lot of strength and are badly affected by it. Caparia will get killed by it. And then some of the blue gums will get killed by Phellinus as well. Eucalyptus ligna, Eucalyptus globulus. But just because it's attacked doesn't mean that it's going to kill them. So it generally is a weak pathogen and a fairly good saprophyte. But they do end up with total death. Phellinus has a rusty color, olivey rusty colored hyphae. Again, it's got these pores on the bottom side. So all of these bracket fungi have pores on the bottom side. And the fruiting body, let me just go back up a couple here. Fruiting body is always more than half the width high. So they're quite big and hoof like. So that'll help you identify them. The reason I say that is the next group are these groups, the Ganoderma. And Ganoderma are quite shallow in comparison, so less than half the width high. And they're often glossy on the top side, white on the underside. Now this one's growing on a palm, and because palms have wall 1 and wall 3, but no wall 4, they can't ever get stronger, they just get weaker. And so palms, fungi growing on palms is a cause for concern and often results in the need to remove the palm. Whereas trees can keep on growing, and we'll have a look again. So that's 3 things, Peter, and let me just quickly make a note. I said white pocket rot. We said, what else do I say? Would show you photographs of, guys?
Brown rot, fungi failure.
Brittle failure, yep.
Yeah, brittle failure, yeah.
And this is the next one. I can't remember what I was going to show you. So next time around, I'll show you some more stuff on fungi. But glossy on the top side, we call this a lacquered look. So these can be quite problematic. White on the underside, this is curry punk. This is on the north of Fagus and Barrington tops. Glossy on the top. Now, it doesn't look shiny glossy, but it's glossy enough. Here it's just because other things are growing on it. And this one, the artist punk, you can draw on the bottom side of it and leaves nice brown stains. Virtually none of them were left unstained. So when I found this one, I was really happy. Everyone likes to write on them. This is one that comes from Queensland. This is a stipulate form. That means it's got a stipe. Stipe is the stem. And if you want to know how you remember these dumb, stupid words, my family still gets tortured when we have mushrooms. I ask them whether they want them stipes on or stipes off. And all my kids know that that's called a stipe. It's just about constant repetition. So this one grows on a root in Queensland, but you can also get a stipe developed sometimes if you've got a very small opening in a wound and you've got Ganoderma. Ganodermas are butt rots. Butt rots decay near roots, and roots give stability. So we always treat Ganoderma more cautiously than we treat white stem rots. White stem rots, Phellinus, can occur to the ground, but it's nowhere near the same in terms of its impact as Ganoderma. Okay, the last one's just a fungus that is an unusual fungus, and it's an interesting fungus, so it's here more for our education than being worried about trees. Cryptococcus neoformans and Cryptococcus gallii, red gum disease. And it's one of those things that generally is not a problem for people unless they're immunosuppressed. Now, if you're an old fellow like me, immunosuppressed means you've got HIV/AIDS. But that's not the case. As you get older, you realize that immunosuppressed means, for example, you're Adam Tom and he's had a heart transplant and has to have immunosuppressants to stop the rejection of his heart transplant. Or you've got like my daughter, and you've got psoriasis, and she takes an immunosuppressant to do with psoriasis. A lot of autoimmune diseases now use immunosuppressants to help. So it's a bigger group, a lot bigger group. In fact, even if you're just a sickling like my youngest daughter, who's almost exposed to everything, you'd have to be more cautious. The great story with this is it's our only known plant nosis. Now we know what zoonoses are because we've just dealt with COVID. COVID, we suspect, is a disease that's gone from bats to pangolins, pangolins to people. Well, this is a disease that comes from plants. And we know it comes from red gums. It was first described out of Egypt. In Eucalyptus camardulensis, it occurs in Australia. We know that older trees are more likely to have it. It can be helped spread around by birds moving things, and that's not to surprise us. But the real breakthrough came from a young lass in San Francisco who at 12 or 13 years of age said to her father, "Dad, I'm bored." And those of you who are parents will know that's the thing you should never say to your parents. So her father, who was an immunologist, gave her a whole pile of swabs and said, "Well, go out and swab something." So she did. She went out and swabbed trees. And indeed, they found Cryptococcus on the trees, and it is a disease of trees. It has almost no impact on the trees. But if you're immunosuppressed, it can cause problems. It can actually kill you, cause problems with your lungs if you're heavily immunosuppressed. But even if you're moderately immunosuppressed, it can cause real problems with ulceration of wounds. And I say this, if you don't like yucky pictures, the next picture is yucky. This is an ulcerated wound from Cryptococcus. So if you've got a wound that's not healing, that doesn't respond to antibiotics, and you are working with trees, or you know somebody in this situation, let them tell the doctor that this is what they're doing with their life. They work with trees, and that may give them a hint to check for a fungi instead. So this is a fungus from trees, and a broad range of trees, not just red gums, that does affect people. I'll make the PDF available if people would like. Let me know, and I'll put that up on the thing.
Question from Ryan, Mark.
Yeah, I see it there. Which fungus forms thick white mycelium sheets between rings, particularly in eucalyptus species? Ryan, I need to see photographs. So, because I'm not sure that I know what you're talking about.
One.
Well, I'm not painting the picture in my head. So when I can, I will, but that's just not giving it. So if you can grab some photographs at some stage. That's what we here at TIPS to do, is to help identify things and sort things out. But I'm gathering it's probably Phellinus, but that's just a gut level. Okay, guys, I'll see you in a month's time or next week if you're doing war as well.